Welcome all, and thank you for joining today's AWRI's webinar. My name is Christian Varela, and I'm a principal research scientist at the AWRI. My background is microbiology. In the spirit of reconciliation, the AWRI acknowledges the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. In this session, we will look at bioprotection in winemaking. But before we jump in and make a start, a couple of quick reminders to anyone who is new to AWRI webinars. If you would like to provide a comment or ask a question, please click on the Q&A button on the Zoom toolbar at the bottom of your screen. Type in your question and click send to send it through. We will be holding the Q&A session at the end of this presentation, but feel free to send your questions at any stage. A reminder also that this session is being recorded and you will be emailed a link to view the recording on the AWRI's YouTube channel. So for anyone who has just joined, welcome. Today's webinar topic is bioprotection in winemaking. I'll start the proceedings today with a short introduction on bioprotection, and then we'll have three presenters from different companies who will tell us how they approach the topic of bioprotection in winemaking. So just to give you a brief introduction into the topic, I think we have to start with asking, what is bioprotection? So bioprotection is the active or passive use of microorganisms to present food or beverages and to exclude other spoilage microorganisms, avoiding the effect of flavors, changing sensory, or the formation of toxic molecules. The OIV doesn't have a definition as such yet, so this is the closest we've got so far. Bioprotection can be considered a specific application of biological control, and biological control not only involves the look or the use of microorganisms, but can also involve the use of insects, for example, on those two photos at the bottom of the screen. But there are a lot of terms used as synonyms: biocontrol, bioprotection, biocontrol agents, biocides, bioprotectants. They all have sort of the similar meaning. Why to use biological control? Biocontrol can reduce the utilization of manufactured chemicals in agriculture, and these would potentially decline the use of fossil fuels and have a reduction in greenhouse gas emissions. Biocontrol agents originate from nature, and they can even be sourced from nature, or they will be identical if they are chemically synthesized. Biocontrol has uses in several industries: agriculture, forestry, amenity, home and garden, and public health. And hopefully I will give you a couple of examples today. Biocontrol also is intended to be non-pathogenic, cause no harms to human, and not actually increase the development of antimicrobial or pesticide resistance. How does it work? There are different modes of action to protect crops from diseases. These include inducing resistance in the plant, for example, against the infections without actually interacting with the pathogen at all, competing for nutrients and space with the pathogen, directly invading and killing fungal or bacterial structures or cells, and also producing antimicrobials, which will inhibit the growth or the effects of different pathogens. One of the first biopesticides was the use of Bacillus thuringiensis, or BT, against flower moths, and this was done in the early 1920s. From there, a lot has been done, a lot of research and resources, and currently there are thousands of different BT strains targeting different pests. So bioprotection started quite early. When we look at the research over time, we can see that the number of scientific publications on bioprotection has been increasing exponentially up to today. But when we look at bioprotection in winemaking, this has just started in the last couple of years, and only a few papers are published nowadays. But this could provide exciting developments in bioprotection in general, but also for winemaking. So a couple of quick examples. Bioprotection can be used to protect fruits, and you can see different fruits from cherries, limes, and lemons that are protected by using different strains, Saccharomyces, Arabidopsis, or Cryptococcus, and this protects from infection from fungal diseases. As I mentioned before, biocides are also natural molecules that can protect or can be used as bioprotectants. And this is a quick example just to show you in this case, is a combination of chitosan and catechin that those two molecules can be used to protect from two different pathogens, as you can see there on the second and fourth columns, and they can extend actually preservation time post-harvest. Bioprotection can also be used to protect juice processing lines, and on your left, you've got a picture of a processing line with fungal biofilm in it. This is actually inside the processing line for juice. On the right, you've got the same processing line treated with natamycin and farnesol, two biocides, and they actually eliminate pretty much all the biofilm, keeping the processing line clean. Bioprotection is also used in meats, and this is an example of using lactic acid bacteria to extend shelf life in fish burgers. As you can see there on the left, the package is quite bloated, and this lactic acid bacteria on the right, on the B panel, you can see that this doesn't happen. It's protecting from developing that bloating spoilage, and this treatment actually extended shelf life of these burgers for up to 12 days. Something probably quite different is the bioprotection of ancient stones. And this again is the use of biocides to control moss. And moss can actually grow on stones, can damage stone integrity, increase water penetration, which eventually will have big effects on the stability of the rock themselves. As you can see on the top panel, we've got the blank there in triplicate, and on the second panel below, we've got the treatment with one particular biocide that actually inhibits the growth of that moss on rock. So where can we be using this? You might be wondering. This could be used to protect old castles or old cities like in Cambodia, and also stone statues like the giant Buddha in China. So it's got a bit of, a real, I think, world application. Finally, another interesting application of bioprotection is the use to protect concrete. Yes. So this is actually using a particular strain to deposit some precipitate that actually heals the concrete from any damage. The panel on your left, A panel, shows this strain growing, and the other panels from B to F shows pictures at different time points of the concrete with a crack in it in panel B, and then we can see how the growth of this particular strain starts healing that crack and protecting or recovering that concrete, which is panel F. So hopefully you saw a brief idea of what bioprotection can be used for. And I'll finish with just a couple of take-home messages. So bioprotection is seen as a green and organic alternative to protect food and beverages against microbial spoilage. The main advantage is to decrease the use of chemicals and avoid developing resistance. This is a topic that is attracting a lot of research interest, and there are several applications to different industries, as you can see there on the picture. So now, it's a great pleasure to welcome Alana. Dr. Alana Seabrook has worked in the wine industry for the past 20 years in winemaking, wine research and application, wine microbiology, diagnostics, and analysis. After completing a winemaking degree at the University of Verona, Italy, she came to the University of Adelaide, where she completed her PhD in wine microbiology. Alana has been working as the technical manager for Lafort Australia since 2017, as well as managing technical communications for the Wine Check Laboratories Group from January 2022. So if you are ready, Alana, I will hand over to you. Thanks.
Thank you, Christian. Can you hear me? And can you see my presentation? Okay, we're off to a great start. Good morning, everyone. I'd like to thank the AWI for hosting this webinar and for the opportunity to speak. I'm also grateful to be presenting alongside Evelyn and Duncan, both who I have great admiration for. So I'd like to start by presenting a slide that was presented in the AWITC on Lachansia thermotolerance. This graph outlines the strain variation present in Lachansia thermotolerance that goes into the selection of a single strain for commercialization. It demonstrates that Lachansia thermotolerance can metabolize anywhere from 2 grams per litre of lactic acid to over 12 grams per litre of lactic acid. I cannot stress enough that when we're talking about species in the context of bioprotection, the level of strain variation is enormous. I would encourage everyone to talk to the manufacturer of the strain they are intending to use, as they will understand the strain characteristics better than anyone. So according to Lafort, the definition of bioprotection is the use of selected microorganisms to colonize the medium and limit the development of microorganisms in the pre-fermentation phase. There are many species in the must ecosystem, some of which have a positive and negative characteristics. 2 of which have been identified as having beneficial characteristics are Meshnikovia pulcherrima and Turullospora delbrueckii. Both are extremely cryotolerant and poorly fermentative, which is a benefit. Further to that, Meshnikovia has also demonstrated to be very good at consuming oxygen. Here we show the strain Zymophylla cure, which is, and we've got the OCR, which is the oxygen consuming rate. This could be ascribed to the crabtree negative status of this species, allowing for partial respiration in the presence of high sugar concentration, contrary to other species such as Saccharomyces cerevisiae, unable to respire in grape must. In 2014, Albertin demonstrated that when Hansennia spora was available to or able to proliferate, it was correlated to an increase in VA production, as well as to a prolonged duration of alcoholic fermentation. In the same study, it was found to be the only microorganism present whereby a small addition of SO2 was actually beneficial for its growth. So effectively, by adding a small amount of SO2, a winemaker is unknowingly enabling for the proliferation of Hansennia spora virium. So in 2020, Johnson found that a number of commercial strains used for bioprotection found that the combination of Turullospora delbrueckii and Meshnikovia and Zymophylla rigide had the impact of population reduction for Hansennia spora virium and a corresponding VA decrease. So what about bacteria? If we now look at the bacterial populations relevant to the winemaking process, lactic acid bacteria were not impacted, while the population of acetic acid bacteria was demonstrated to reduce the population, to be reduced over time. So one hypothesis for this is actually the oxygen consumption work highlighted previously that may create a limiting environment for aerobic species such as acetic acid bacteria, and hence may have an impact on reducing the population just by the presence of bioprotection using Meshnikovia pulcherrima. So we work with strains at Lefort. We work with strain of either a combination of Tarullospora delbrueckii and Mechnicovia called Zymaflora rigide, or we work with Mechnicovia alone in a strain called Zymaflora kio. We're working with a dosage rate of 25 ppm generally. So this is 1/10 the rate of what you would normally inoculate a yeast into a ferment with. So we're asking it to do something different. We've been working on equipment as well as must itself and looking at the use of bioprotective yeast to occupy the microbial space on surfaces that would normally only get a rinse during vintage. We've focused on grape transport in Australia, which I'll detail a little bit later. We're really lucky to be working with Adelaide Uni. We've got a couple of very keen honours students who are working on juice storage and extension of the transport work that I'm going to talk about later. And we're also really grateful and lucky to be working with WineCheck Laboratories, who have kindly developed qPCR assays for all the positive and negative strains involved in this process, so we can monitor them by qPCR in real time. So I'm going to talk about two main sort of projects. One was in the US in 2019. We were just talking about this as a picture of a guy with a spray gun. So basically the winemaker said that his harvester smelt like VA. And so we wondered if we could actually occupy the microbial space of surfaces using bioprotection yeast. And so in this study, he treated harvesting machines during over a number of periods during vintage. He conducted micro qPCR analysis by sampling the harvest reception bins, the first 3 bins every day, sampling in the middle of the bin. And so the same sampling process was used in the first harvesting week where it had not been treated with bioprotection. And what he saw was what we've been seeing in the lab and what we see consistently. And that's a reduction in acetic acid bacteria over time. So after progressive treatments of the harvester, they generally saw a reduction in the amount of acetic acid bacteria, and they also saw a reduction in the amount of Hansen sporovirum, simply by occupying the microbial space. So in Australia, we thought we really need to look at grape transportation because we do a lot of it in Australia. We transport machine harvested fruit very, very long distances. And so we looked at a number of trials. This is a very short snapshot of what we did. We looked at several trials where we could have complete control with known distance amounts. And we found that the actual transit times were much, much greater, which we would expect, but some of them were extremely higher than expected. So in this particular trial, all the bins were machine harvested. The bins were treated with Zymaflora rigide. So that's just by spraying it with a weed killer sprayer. So it's simple rehydration, mixing it together in triplicate. So the bins were literally sprayed before the fruit was harvested into it. Several hours prior to picking, there were 9 to 15 bin trials per and 3 to 4 variants per trial. Bins were sampled directly with a sterile sampler prior to processing at the winery at the crusher with a sterile sampler, and these were all cultured for yeast and mold, acetic acid bacteria, lactic acid bacteria, non-Saccharomyces. General observations for these particular trials were that bacteria were not really significant at this early stage. Fruit was sound in all instances, and that all cultured samples were identified with sequencing data through Charles River Laboratories. So in all of these trials, we really needed to understand the best application mode. And we found that if we sprayed it on the bins, we got excellent survival rates of these species. But we could also add it dry, but there was a reduction in population. And if we did want to add it dry, how much did we need to add? So we needed to understand dosage rates. And generally speaking, across all of our trials, we found that if we added double to triple the amount, so 50 to 75 ppm as opposed to 25 ppm rehydrated, we got very similar levels of viable population. And in terms of SO2 tolerance, we do promote the reduction in SO2. We're not saying no SO2, and we don't expect winemakers to transport grapes across the country without any SO2. And in this case, we used 100 ppm of PMS, so 50 ppm of SO2 approximately, and we found good survival rates of these species. So I'm only going to show you details of one trial, and this was the really long one that went to the hunter. And this was done with 50 parts of SO2 as described previously. The truck actually broke down, so it ended up being a 58-hour transit. We didn't know if we were going to get any results out of this. But what we found was, much like we've seen until now in all the lab results and all the trial results, was that the Meshnickovia pulcherima and the Turulaspora delbrueckii managed to outcompete everything in the bins where they were actually inoculated or sprayed. And then in the control bins, we see this proliferation of indigenous Saccharomyces yeast and also Hansen sporovirum, which is in greater proportions than the actual Saccharomyces. So just coming back and trying to conclude, for us at Lefort, bioprotection is really about using microorganisms that are already present in the grape must to colonize the medium and represent the majority of detected microflora, and then also limit the development of other microorganisms, which we saw in those trials in the pre-fermentation phase. With regards to harvesting and cellar equipment, we're able to use non-Saccharomyces yeast species with beneficial attributes to populate the surface of the harvester, and populations of known spoilage microorganisms were observed to be reduced over time. In terms of cellar equipment, hand harvesting bins, conveyor belts, crushers, and presses, we were able to populate them with Turulaspora delbrueckii and Meshnickovia and minimize the presence of mold over time. When we go back to transport of grape bins with machine harvested fruit, the bins were able to be treated up to 19 hours prior to picking and maintain a viable population. In some instances, we had to treat the night before and the grapes weren't picked till whatever time the next morning. Zymiflora gide was able to survive with high levels of SO2 for transportation. In terms of dosage rate, 25 ppm was able to populate the liquid phase in a very consistent manner across all trials with no risk of fermentation below 15 hours of transit. Spraying the yeast on the bins rather than dry application appeared to maintain a more consistent level of yeast in the liquid phase prior to processing. And dry addition is possible, but we recommend 50 to 75 ppm. Turulaspora delbrueckii can start to ferment over time with higher temperatures in very uncontrolled temperatures. But the benefit is it's a known strain, it has beneficial attributes. But we would not, it's very poorly fermentative. So the amount it would actually start to ferment is very, very, very slight. But apiculate yeast were able to proliferate in control situations with only SO2. So with regards to non-sac yeast at Lefort, we launched Zymiflora alpha in 2010. So that's a straight strain of Turulaspora delbrueckii that's designed to actually metabolize. So we're talking about a 300-part inoculation. In 2017, we launched Zymiflora gide, which is a Meshnickovia pulcherima and a Turulaspora delbrueckii strain. So two species mixed together. And the inoculation rate there is more from a bioprotection point of view. So it's 1/10 of that dosage. So 25 parts generally. In 2021, we launched Zymiflora cure, which is a straight strain of Meshnickovia pulcherima, which has excellent ability to consume dissolved oxygen, extremely cryotolerant, and which is why we launched it, and very, very poorly fermentative. I haven't talked much about Zymiflora omega in terms of bioacidification, but it is the last of our non-Saccharomyces yeast in this strain, in this range. And thank you very much, everyone. I will pass over to Evelyn.
Cool. Thank you very much, Alana. That was really, really interesting. Now now I've got the pleasure to introduce Evelyn. Dr. Evelyn Bartowski is Research and Development Manager and Applied Microbiologist at Lallemand, Australia. She oversees all industry trials and research projects with universities and research institutes. With over 30 years experience in microbiology, Evelyn provides fermentation technical support in Australia and New Zealand. And previously, Evelyn led the bacteria research program here at the AWRI. Now, Evelyn, if you're ready to make a start, I will hand over to you. Thanks.
There we go. Okay, so I think you can hear me and you can see my screen. I've had no, great. So I'm going to continue on with bioprotection as a natural approach against microbial spoilage and oxidation. Christian has given a great introduction, and Alana has also talked lots about it. And of course, you know, we know what bioprotection is. It's the use of microorganisms to preserve foods, beverages, and really to exclude those other microorganisms and thus avoiding off flavors. So why are we looking into this? And of course, you know, we know that SO2 is a traditional way to avoid contamination and oxidation of wine. We are seeing a lot of consumers asking for less chemical additions, hence seeking alternatives to SO2. And as Alana said, we're not looking to eliminate SO2, it's simply a way to reduce the usage of it. So one of the biggest challenges in winemaking is to reduce SO2 while preserving wine quality. Oops. So bioprotection is one of those best alternatives to SO2. There's an emergence of a lot of non-Saccharomyces, but we need to remember that there are a lot of genera and species, and not all species are equivalent, and also not each strain within a species is equivalent, such that each strain has its own unique properties. I'm going to specifically be talking about the Metchnikowie species 2, Fructicola, and also Pulcheremia. So this genera, or genus rather, has very interesting enzymatic activities which can also impact wine aroma, and I'm not going to speak about that aspect of the Metchnikowie today, but they also have strong biocontrol activity against various microorganisms, and it's this aspect that we're going to look at. So bioprotection mechanisms, there's a variety, basically the indirect ones where we're looking at competition for growth, and also direct methods through antimicrobials. So the indirect, by competition for nutrients and growth space, is one way and is very efficient, and also using antimicrobials. So for example, in the Metchnikowie genus and the strains, they're able to produce pulcheremia. You can see that here. It's often referred to as the pink yeast or such because it has pulcheremia production and has these red halos, so very easy to distinguish or identify. And also a common theme that we will hear is that there's a lot of strain variation in their abilities to do things, including in the production of pulcheremic acid. So if I come over to Metchnikowie fructicola, so this is a strain that was actually isolated from apples. Christian alluded to how well these non-Saccharomyces or biocontrol can be in all different kinds of fruits, not only used in grape production. So its protectant works, it's not a fermentative strain, it is one that takes up space, and also it is used, this particular strain that we have in Lallemand is recommended for pre-fermentation during cold soak. They're very cryotolerant, so they do well at low temperatures, and we use it as a means to reduce the SO2 usage. So in the left bottom graph, we can see how by using biocontrol with fructicola is able to really reduce the amount of VA, acetic acid, that is produced by Hansennia spora. So we can see that great reduction there. And on the right on the graph, we can also see how the Mechanicolia fructicola is able to minimize or reduce the evolution of acetic acid bacteria, another source, of course, of VA. So you can see the growth of the acetic acid bacteria in red, and with the addition of the fructicola strain in green, you can see how that population has really reduced, and the blue line is showing the population of the fructicola strain. So another example here in as a managing contaminants in cold soak and by control of specific non-Saccharomyces. This is an example in a Merlot, and we can see how fructicola has been able to reduce that yeast population during the cold soak process. So this has been done for 4 days at around 10 degrees. And we can really see how the population of the Cerevisiae has been reduced, and the non-Saccharomyces. Another strain that I'd like to talk about is a Mechanicolia pulcheremia. So Alana has already alluded to the ability of these species to absorb oxygen. So taking advantage of this concept, particularly in the production of white wine and rosé wine making, we worked with Vincent Gerbeau in Burgundy, looking at selecting a strain for its high ability to consume oxygen. So screening over 500 different strains, we were able to identify a strain that was able to consume oxygen at a very, very high rate. And we can see this in 3 different wines in the bottom, in a Sauvignon Blanc, a Grenache Rosé in the center, and a Riesling on the right. And the Mechanicolia strain is the yellow strain with the arrow there, and we can see within a day all of the dissolved oxygen is gone following inoculation. So benefits is bioprotection, so it's able to control the growth of various microorganisms. Here I've got an example of acetic acid bacteria, and we have the control in orange at the top. You can see the growth there over the period of time, and in the presence of the pulcheremia strain, we can see that there is a lot less growth of the acetic acid bacteria, and at the end of the fermentation, we can see a dramatic decrease or difference in the volatile acidity, around 0.3 with the pulcheremia strain in this particular example, versus without using bioprotection, we've got 0.55 grams per litre of VA. It also protects beautifully against oxidation. So this example here I'm showing is in looking at the dissolved oxygen after pressing, and in fact doing this as a comparison to sulfur dioxide, no sulfur dioxide, and the pulcheremia strain. And we can see on the graph on the left that there's a lot less dissolved oxygen, equivalent to what we see with the sulfur dioxide. And on the right, we actually can see it visually. So what we see here is that the pulcheremia strain is able to manage that oxidation in a very equivalent way to SO2. Also, we see an impact on the aroma compounds. Here we're looking at Grenache Rosé and also a Sauvignon Blanc on the right, and looking at those volatile thiols. In this case, I'm just showing a graph with the 3MH. And we can see a control with SO2 and with a Pulcheremia strain. We can see that there's much better preservation of thiols in that final wine using bioprotection with the specific Pulcheremia strain. One of the other benefits that we have been able to find with the Pulcheremia strain that we have is its ability to reduce copper content. In fact, it can reduce the copper content in juice, and so that this is also a complementary mechanism of bioprotection against oxidation. So you've got that fast oxygen consumption and also copper removal. I'd just like to give an example of Sauvignon Blanc trial this year in Western Australia using Parongarup Sauvignon Blanc from the Great Western. And here we were looking to demonstrate and see the impact of bioprotection during cold stabilization of the Sauvignon Blanc by using the Pulcheremia strain. And we can see over 10 days in this graph here. So we've got dissolved oxygen measurements. The control is in the yellow bronze and the Pulcheremia strain is in aqua. And we can see the decline of dissolved oxygen over the cold storage over 10 days. And in fact, after that period, there was 61% less dissolved oxygen in this juice with the use of Pulcheremia strain. So bioprotection using unique Mechanicolia Pulcheremia strain is a great alternative for SO2, particularly to help reduce it with its anti-spoilage and antioxidant properties. So it has good survival of low temperature, produces pulcheremic acid to help against spoilage. I've hoped to show that it has fast consumption of oxygen, abilities to remove copper. There's no fermentation and has low nitrogen requirements. So it's a great protection of must juice color and also protection of aroma compounds. So bioprotection is a natural approach against microbial spoilage and oxidation. Unique Mechanicolia strains with specific bioprotection properties for red and white winemaking. So the two strains that I was talking about today, the Fructicola strain is in the Lallemand range as IOC Gaia and is great for the protection of red grape must during that cold soak maceration. And the Pulcheremia strain that I described and talked about is in our portfolio as level 2 Initia and gives lovely protection against oxidation, also microbial protection at low temperatures during stabulation and storage of juice. So thank you very much for your interest in bioprotection, and I look forward to answering any questions afterwards. Thank you.
Thank you very much, Evelyn. That was a very interesting presentation. Now, I would like to introduce our next speaker, Duncan Ham. He's senior applications scientist working within the fermented beverages team at Christian Hansen head office in Denmark. He studied chemistry at the University of Waikato in New Zealand and then worked for 10 years in the dairy industry, including 5 years with Christian Hansen. After completing a graduate diploma in viticulture and enology in New Zealand, Duncan rejoined Christian Hansen as a commercial enologist in 2008. Before taking up his current role in 2021, he was based at the Melbourne office of Christian Hansen for 5 years. So now, Duncan, if you're ready to make a start, please, yeah.
Thanks, Christian. Thanks for that kind introduction, and thank you for everybody to come in and listen to what we're talking about today. The topic that I'm going to be covering is actually Lactobacillus plantarum, or Lactobacillus as it's now known, Lactobacillus plantarum for biological protection in winemaking. So by way of an introduction, we've heard we've seen some really great data from Evelyn and from Alana on using non-Saccharomyces yeast for biological protection. But we want to pose a question: is there a place for lactic acid bacteria as well? And in Christian Hansen, we work a lot with yeast. We work a lot with non-Saccharomyces yeast, but we also see that there's this absolutely great potential for lactic acid bacteria. And I've lifted this table out of a really good literature review that was published just a couple of months ago by the University of Turin. It's a great reference to get the latest update on the field of biological protection. And actually, they talk a lot about lactic acid bacteria, not just yeast. So in Christian Hansen, we have one particular strain that we've had in our strain collection for some time, and it's a Lactobacillus plantarum. In our internal strain collection, it has the number 12399, but it's also being registered with the German state microbial depository and has a DSM number of 27565. So I'll use that reference for it for the rest of the presentation. But to give you an idea of what we've seen, the two photos that we can see are where we've plated samples. On the right, they're grapes. On the left, it's some juice samples. But for each of those agar plates, the one on the left-hand side is untreated. The one on the right has been treated with this particular strain. And it's really clear. You can see that while there's a little bit of mold growth, it's pretty well controlled against the untreated sample. However, what about using bacteria or this particular strain to control issues that arise from, say, acetic acid bacteria, or perhaps even rogue Unococcus that could cause unwanted MLF? So we set about to look at that and to investigate it. And this is just some examples of some initial bench investigations. On the left-hand side here, we have some results where we took heat-treated air and juice. It had starting malic acid of 4 grams per litre, and we inoculated with 2 different strains of Unococcus. And we also inoculated with the same 2 strains of Unococcus in combination with the plantarum. Now, the level of Unococcus we added was 10 to the power of 5, so it's 1 cycle lower than what is the critical population to conduct MLF. And we went in with 1 log cycle higher of the plantarum. And what's really interesting, on the left-hand side, we have the malic acid concentrations at 3 days. And at this point, MLF is almost complete in the samples that just have the Unococcus. And by 14 days, it's absolutely complete. But what we also see is the effect of just take my pointer off. We see the effect of this particular strain is able to block the MLF when it's present. So that's very, very interesting. That is at bench scale. But another facet we looked at bench scale was inhibition of acetic acid bacteria. And so this is also a challenge test. We took a grape juice. We added 1% ethanol to it, and we actually contaminated it with 1 million cells of acetic acid bacteria. But we had 2 treatments where we treated with 2 different strains of plantarum, and we effectively incubated all 3 treatments over 3 days at 25 degrees. And what's really interesting is that, first of all, in the control sample, we see a 2.5 log increase in the Acetobacter concentration. But if we compare that to the one that's been treated with the DSM 27565, we actually see a decrease. And in fact, the difference between the two is almost 4 logs. So there's a very, very clear inhibitory effect against acetic acid bacteria. But what happens if you take it to a bigger scale? And this is where it becomes more relevant. And so I just wanted to introduce some trials that we ran at Melbourne Polytechnic's winery in Ararat in 21 and 22. In 2021, we used Cabernet Sauvignon. In 22, it was Shiraz. But we used the same experimental design for both years. We had 4 different treatments, a run and duplicate with around 200 kilograms of grapes per tank. And we either added the experimental plantarum culture or we added tartaric acid. We also had a little bit of SO2 added, so 20 milligrams per litre total in 21, 25 and 22. So just to give an overview, there's 4 columns. The first column has tartaric acid and no plantarum. The second column here does have the tartaric acid and the Lactobacillus plantarum. The third column has no treatments, while the fourth one is just the plantarum by itself. So I'll just give you a little bit of a snapshot. We did extensive testing both years, and both years the sensory on the samples treated with the plantarum was clearly superior. But just to look at some microbial results, on the left-hand side here we have some qPCR data from 2021, and we actually ran an assay where we had a generic probe for Unococcus ini, so it's not strain specific. But what's really interesting is that in the third sample along, we have quite significant outgrowth of Unococcus. Now, this is a sample that's not had any acid and it's not had any plantarum added to it, and it has the most growth. But what we also see is both the acid and the plantarum are good at keeping the Unococcus suppressed during primary fermentation. In fact, the one that where it tends to be the lowest is actually where it's the plantarum at the higher pH level. And that also makes sense because plantarum tends to like a slightly higher pH than Unococcus does. One comment around this is when we present this data, a lot of winemakers will then ask us, will this stop me having a spontaneous MLF down the line? No, it won't. It is literally there just to inhibit MLF during primary fermentation. The next set of data we have is from 22, and we actually looked at the acetic acid concentrations over time. And generally the grey and the blue are the two with the DSM 27565 culture, and they generally have a little bit lower concentrations throughout the trial. However, the most dramatic difference we saw was once we'd bottled the wines and analysed them, it was significantly lower concentrations of acetic acid in the one with the experimental plantarum, particularly the one that had the higher pH where the plantarum can really flourish, which also suggests it's actually not just something that can replace a little bit of SO2, but can replace a little bit of tartaric acid, or might fit in with varieties that tend to have a high pH, say Tempranillo. So this is my last slide, and I just wanted to put in some suggested further reading. In the last couple of months, there's been a paper published by Engelsos and others from the University of Turin looking at this particular strain against acetic acid bacteria, Hansenia ovarum. So it's well worth consulting. It's published by the American Journal of Viticulture and Enology. And there was also a poster one of my colleagues had accepted at the Tech conference this year in Adelaide. And last of all, I just wanted to talk about the commercialized culture. Last year, we commercialized this strain under the brand name Viniflora Nova Protect. It's one bag designed for 3.5 tons of grapes, or 2,500 litres. It can be added in the winery, as the picture on the left shows, or it can actually be added in the vineyard as well. And the picture on the right is actually from Pathogenic, in a trial conducted last year for a winery in the Riverina, where we basically sprinkled the culture into the bins before picking the fruit. And it's extremely successful trial. The wines were very clean, and actually the ones that were preferred by the winemakers were the ones that not treated with sulfites, but treated with the Nova Protect. So thank you very much. I think I might have gone about 1 minute over my time, and yeah, would welcome any questions.
Thank you very much, Duncan. You're done exceptionally well considering the time difference. For everyone who doesn't know, Duncan is around 2 to 30 in the morning at the moment in Copenhagen. So yeah, thank you.
2:52.
Yeah. Cool. So now we got the opportunity to have a Q&A session, and you can ask questions to any of our presenters. So still waiting to see any questions. So I'll probably make a start. Alana, on the transportation trials that you presented to us, did you guys have a look? I know that is the intention to have a look at what's going on during transport and maybe storage, but did you have a look at if those trials had any effects on fermentation kinetics, on sensory later on after fermentation?
So in terms of the transport trials, we had a lot of limitations around taking samples and working with wineries during vintage at a very busy time when you're dealing with fruit intake. So we made a point of taking them from the bins. We weren't able to separate them off, but in the situations where we did, so I listed a lot of trials that we did there that I didn't talk about, and a lot of the we've done a lot of work on whole juice transport as well, and we see very similar results to what Evelyn actually talked about in terms of the aromatic profiles are greatly lifted whenever we use definitely Zymiflora Gide, not so Cure. I need to see trials on that, but I imagine it would do similar things. And Cure, when we use it in, so the Meshnikovia, when we do it in stabulation, we certainly see differences in terms of aromatics as well. So not so much for the transport trials, but certainly for juice storage and juice transport, yes.
Cool, thank you. We've got a couple of questions coming. There's a question about, is there any post-fermentation bioprotection products?
I think Duncan's the closest to this one. At this stage,
yeah.
I mean, yeah, you go.
Oh, sorry. It depends if you include malolactic fermentation as being post-fermentation. We do see, particularly in markets like France, where we have a lot of customers making preservative-free wines, using one particular Enococcus we have, which is citrate negative, so it doesn't produce any diacetyl, as quite a good tool. Putting wines through malo, which wouldn't necessarily go through malo, just to remove a fermentable substrate, but not having any malo flavors. But it's once you go after fermentation, I think it's more challenging because you've got alcohol, you're really limiting what's going to survive.
Yeah, so we take very different levers to try and protect post-fermentation, including yeast, sort of yeast extracts, and to sort of scavenge oxygen, and also chitosan-based products to try and sort of limit populations. But again, it's exactly what Duncan said, to try and find something that survives in those conditions is quite a challenge.
Exactly. We take the same approach in inactivated yeast type products to manage finished wines and chitosan based.
Thanks. Probably since you are the Evelyn, there's a question about the ability of some microbial cultures to reduce dissolved oxygen and copper. Do they make them potentially useful to protect against lacasa oxidation in wet years?
So potentially, yes. It's an interesting question because the botrytis infections there, so it is potentially a way to help reduce the impacts. But of course, it's not going to take away any lacase that's there already before it comes in, but it can definitely be used to help minimize the growth of the botrytis from during transportation. So similar to what Alana, she talked a lot about the transportation. The 2 strains that I talked about can also be sprayed into bins to give the protection already from the point of harvesting the fruit as it is transported to the winery.
Cool, thanks. Probably this question is mainly for Alana, but anyone else can also answer it. So it's from John Summers. How quickly, I guess, these products being taken up by vineyards shipping grapes?
Sorry, I didn't understand the question.
So I think, I guess vineyards shipping grapes, how many, or the demand of using these products in your, I guess.
Transporting grapes?
Yes.
I think it's like anything, it's a new concept, it's an education piece, and it's about doing the trials and working out the benefit for the winery. Once we're talking about being in the vineyard, so we've got to make sure the process is simple and it's quick. So I think if when we work with a winery, if they see benefit and it helps them, then we get uptake. But it's about the winery systems and whether it fits their process as well. That's why we tried to look at different applications and methods.
We've seen uptake in transportation of juice, for example, as a way to really help minimize using too higher doses of SO2. And again, there, it's so that you don't have really high SO2 juice, which then needs to be processed. And if it's going to go through Malo, there will be lots of issues at that point, because SO2, if it's high concentrations, obviously is going to be inhibitory to Malo. But we have seen good results transporting from Western Australia across the country with these bioprotection products to help minimize them starting to tick.
Cool. Thank you. The next question is from Jeff, and it could be, to any of you guys. Do any of these bioprotectants lead to stuck primary ferments or stuck MLF?
So we have, we haven't seen that it is. Most of the Mechanicaria strains are cryotolerant, so they're sensitive to temperature increase once the ferment starts, and also they're not very alcoholic tolerant. So in that sense, there's no impact on the primary ferment. However, they are proliferating, they do grow, so they obviously will use some source of nitrogen. So you do need to make sure that when you overseed and inoculate with your Cerevisiae strain or preferred strain, that you manage the nitrogen requirements. They do use they will use vitamins, minerals, and we do know that that's important for fermentation. So you do need to be aware and manage some of the nutrition for your Cerevisiae that you inoculate afterwards. But we haven't seen stuck fermentations due to the Mechnicaria strains or Malo.
Duncan, we can't hear you.
Mute.
Sorry, if I was going to add anything to that, the only one that the species that I'd say is the one to watch out for, but it's perfectly manageable if you can control your nitrogens, is Lachantia thermotolerans. And in Christian Hansen, we have 2 of them. That tends to have the closest and the highest nitrogen demand amongst non-Saccharomyces. So that would be the only one to be aware of, but it really isn't an issue if you're controlling your YANs.
But I think, I wouldn't mind just chipping in here. I think it is really important to look at the dosage rate that we're talking about and what we're asking these species to do. So with Lachantia, I totally agree, but we're actually inoculating it, well, at least for us, we're inoculating at a much higher rate, and we're actually asking it to do to metabolize sugar. Whereas with the concept of bioprotection, in the case of the strains that Evelyn and I are talking about, and I'm not sure what levels Evelyn's talking about, but we're talking about 1/10 of the dosage. So we're just asking them to survive. So we actually haven't seen any nitrogen consumption of these yeast species in bioprotection. And we have done a little bit of work on botrytis. And I think where we saw the greatest benefit, we still need to do more work on it, but where we saw the greatest benefit was the reduction of the associated acetic acid bacteria populations with that. But again, as exactly like Evelyn said, it's not going to protect you from lacase activity.
Oh, thank you. Just following that up, Alana, since you mentioned dosage, and this is a question for anyone, is the cost barrier for using bioprotection products, at low application rates, is a good balance between the cost and benefit of using them?
A packet goes a long way.
Absolutely. And it's also the whole concept of understanding lower chemical use, looking at alternatives to SO2. So if you're looking at organic type of wines or preservative ones, you're minimizing your use of SO2 and chemical inputs, and you're preserving wine quality. So all these aspects need to be balanced in when you decide, if you're thinking, okay, I do need to buy 2 yeast for this, I need to buy 1 for bioprotection, and I need to buy 1 for inoculation. But ultimately, you should balance that with the quality of the wine that you're getting at the end.
And I'm not sure if you've seen this, Evelyn, but I'm sure you have. Whilst we don't, we can't talk about wild fermentations because we don't have a lot of control over them. We do have quite a few people using bioprotection in wild fermentations for the simple fact that they get a much cleaner fermentation.
Exactly.
We know that the non-sacs can't take off, and the fermentation kinetics of Saccharomyces cerevisiae will naturally outcompete them. But they're getting much cleaner ferments at the end and much lower VAs. So they just like the security of a clean ferment, but using the yeast that's in wherever.
Yeah, absolutely. So the bioprotection isn't killing the indigenous yeast that you might use in a wild ferment. It's simply suppressing their growth. And so as the temperature, as you might increase the temperature, for example, in a cold soak, you won't be fermenting at 4 degrees. Rather, as that temperature increases, the Mechnicaria and strains and products that Alana spoke about as well, they are cryotolerant. Hence, they will not. They will slowly die off, and the indigenous population take off. And as you said, Alana, it's just cleaner at the end.
Cool. Thank you. A question for Duncan. Inhibition of MLF during fermentation, has this been a widespread problem?
I would say when I was based in Australia, it was quite rare, but in Southern Europe, where tends to be typically high pH and must, lower level of hygiene, and more of a commitment to running wild ferments, it can be a serious problem in some regions. Yeah, not necessarily in every cellar, but when it happens, it's terrible because it just leads to high VA. So if we can help solve that, then it's a really good win for those cellars that tend to struggle with it, and the ones that do tend to struggle with it year in, year out, in our experience.
Thanks.
Yeah.
We've got one last question about nitrogen requirements. Is there a maximum concentration that bioprotective strains will reach, or they will keep on reproducing and growing until all the nitrogen is consumed?
No. So, I mean, the thing is, we have to remember like I said before, we're inoculating them, but at a super low rate. We're not actually asking them to do anything. So, and I think even if we put these, if we're talking about both Torulaspora delbrueckii and Meschnikovia, we're talking about extremely slow fermentation kinetics, which is one of the benefits of them. And so if they were to ferment in ideal conditions, I mean, what would you get, Evelyn? 1% for?
Yeah, absolutely.
1% of alcohol?
If you're lucky, exactly. Yeah. So
Torulaspora is a bit higher, but
yeah,
it's still very slow.
Yes, and we're not talking about massive proliferation. They're just taking up space. They may multiply once, possibly twice. And so the amount of nitrogen and those micronutrients consumed is not massive or anything. So small amounts. But good practice is why I mentioned earlier that you should make sure that you are doing the nitrogen nutrition management of your inoculating strains.
And correct me if I'm wrong, Evelyn. The literature, the scientific literature actually says Meschnikovia and Pichia cluveri, which we have, actually, once you're halfway through fermentation, nitrogen actually starts to increase again as cells lyse. So well, they actually put nitrogen back in some cases.
Yes, absolutely. So as we've been saying, they're alcohol intolerant. So within a few degrees, or once you get up past 5, they're definitely dying off, and that autolysis will do exactly that. Not that it's replacing nitrogen nutrition management, but they are contributing.
Yeah, exactly.
Great. Cool. Thank you. I don't think we've got any more questions. So with that, I would like to thank our 3 speakers, Alana, Evelyn, and Duncan, for providing very important insights into bioprotection in winemaking. I'm sure everyone who attended today got a lot out of this session. I would also like to thank you, the audience, for joining in and taking part. And I would like to remind you that you will receive a link to view the recording of this presentation later on. I would also like to acknowledge Wine Australia for providing funding and support for the webinars via the AWRI extension project. So thank you again, and I look forward to seeing you at the next AWRI webinar.
Thanks, everyone.
Is there.
Thank you.
Thank you very much.
Thank you.
Cool. Thanks, guys.